Hi everyone, I'm Jackie and I'm here to show you electroporation using the neon transfection system. Specifically, I will be transfecting induced pluripotent stem cells for CRISPR-Cas9 genome editing. So before you begin, you want to make sure that your cells are looking healthy and you want them at around 70 to 90% confluency in your culture dish. So I have a 6-well plate containing my hiPSCs. I'll show you on the microscope about what they should look like. You can see that they're about 70% confluent or around there, and the cells look good, so we're good to transfect. So to prepare your cells, the first step is to dissociate your cells from the culture dish. To do this, you want to first aspirate the media from the cells. Next, you want to rinse your cells with PBS without calcium or magnesium. You can rock your plate back and forth a little bit to make sure that all the cells are rinsed, and then you can aspirate the PBS. So now you want to add your dissociation reagent. In this case, I'm going to be using Triply Express. This is a really good enzyme used for dissociating a wide variety of adherent mammalian cell types with minimal damage to your cells. For a 6-well plate, you can add about 1 ml of media into the dish. Now once your Triply Express is on, you want to put your plate into an incubator at 37 degrees Celsius for about 3 to 5 minutes, and this should allow your cells to lift off of the plate. So while the cells are dissociating, I'll go ahead and tell you a little bit about the neon transfection system itself. So there are 3 main pieces to the device. One is the neon device, which is this silver benchtop piece right here. It has a really easy to use interface, which is really nice, and it has a touch screen. It also comes pre-programmed with a 24-well optimization protocol. If you're looking to determine which protocol is best for your cell type, there is an online neon cell database, and it has recommendations for the protocols for most common cell types. So I would check that out if you don't know which one to use. The second piece is the neon pipette station, and this holds the neon tubes. These tubes come in the neon kit, and they have an electrode at the bottom, which actually transfers the electric field from the electrode in the neon tips. The neon tips come in 100 microliter and 10 microliter size, and for the case of the training today, I'm going to be using the 10 microliter tips. I've also already loaded my neon tube into the neon pipette station, and I have added 3 mils of buffer E, which also comes in the neon kit, into the tube. The third piece is the neon pipette, and this is a fixed volume pipetter, and it's designed specifically for the neon tips. And when you're ready to transfect, you load the entire pipette into the neon pipette station for the transfection. So our cells should be done dissociating soon, so you can now prepare your plate that you will be putting the electroporated cells into. I have pre-coated a 24-well plate with laminin 521. This is a feeder-free culture matrix that, when used in combination with our StemFlex media, will give you really good post-electroporation cell survival. StemFlex in general is really good for any application where your iPSCs are under stress, particularly if you're using single-cell passaging or doing reprogramming, or in our case, genome editing. And then to further enhance post-electroporation cell survival, I'm going to add Revitacell into my media. And Revitacell is just ROCK inhibitor with additional antioxidants, and this is really good to use whenever you're singularizing your iPSCs. It'll give you really good post-electroporation survival. So I've already prepared the StemFlex media with Revitacell, and I'm just going to add a ½ ml of media per well of my 24-well plate. And you want to prepare as many wells as you have transfections to do. Since I'm just demonstrating today, I'm just going to prepare one well. So you just want to aspirate off the laminin 521 that's already on there. And then add a ½ ml of your StemFlex plus Revitacell. So now our destination plate is ready and our cells should be dissociated. You can check to make sure that your cells are starting to lift off the plate by looking at them under the microscope. You can see that the cells have started to round up and they look like they're starting to lift off the plate. That's a really good sign, and your cells are ready to harvest. So to neutralize the triply express that we put on the cells, you want to add 1 volume of your culture media to the amount of triply express that you have in the well. So I'm going to add 1 ml of my StemFlex plus Revitacell on top of the triply that's in the well right now. And then you can rinse your well a couple of times to make sure all of the cells come off of the plate. And then you want to collect all of your cells in a 15 ml conical. So at this point with the cells that you harvested, you want to get a cell count, and then you want to spin your cells down at 300 Gs for 5 minutes. And then you want to aspirate off the media and triply, and then resuspend your cell pellet in buffer R. Buffer R comes in the neon kit as well. And you want to resuspend your cell pellet to a density of 20 million cells per milliliter. So for the sake of time, I have already prepared my cells at 20 million cells per mil. You want to make sure that your cells are singularized, because this will be the best for transfection and give you the best transfection efficiency, so I'm going to just pipette my cell suspension a few times to make sure they're still singularized. So I have also pre-prepared a tube that contains my guide RNA and the Cas9 protein. The guide and protein have been pre-complexing for a little over 10 minutes, and you want to make sure that they pre-complex for at least 10 minutes at room temperature before you add the cells to it. And then at this point, since it's already been pre-complexing for 10 minutes, I can add 10 microliters of my cell suspension to the guide and protein pre-complex. And now your cells are ready for electroporation. So the first thing you want to do is make sure that the neon pipette tip is properly attached to the neon pipette. To do this, you want to take the pipette and depress the plunger all the way down until the clip at the end of the pipette is open. And then you want to put a good amount of downward force onto the neon pipette tip and then just release the plunger. And then your tip should be properly attached. Then you can take your cells plus payload complex and just pipette up 1 pipette volume. Try not to get any air bubbles in there because that could affect your transfection. And then you want to just put the entire pipette with the tip and your cells in it into the pipette station. And make sure you push all the way down until you hear it click into place. Now if you go over to the neon device, you can select your protocol by pressing the optimization button. And then all of the protocols will pop up and you can select the one that you want to run. For the sake of this training, I'm going to select option 5. And then I'm going to press load to load the protocol. You can see that protocol 5 is loaded up here. It displays the number 5. And then all you have to do is press the start button and the electroporation should take place. Sometimes it displays a tip connection error. In that case, you might have an air bubble or you might need to just try pulling the pipette out and putting it back in. So let's see if I can get this error to clear. Nope. Let's try this again. So it looks like I do have a little bit of an air bubble. That's probably what's giving me the error. Alright, sorry about this. Let's try it again. So I'm still getting air bubbles, and the issue could be that the tip is not properly attached to my pipette tip, or to my pipette. Hopefully this resolves the issue. Nice, okay, it worked. So once your electroporation is successful, complete will appear on the display. And at this point, you can deposit your cells into the well that we prepared earlier. So that error was likely due to the tip not being properly attached to the pipette. And then you can just use the pipette as you would a normal micropipette and deposit your cells directly into the well. Then you can eject the tip by pressing all the way down on the plunger. And then you can rock your plate back and forth a little bit to make sure that the cells are evenly distributed throughout the well. And then I can show you on the microscope what the cells will look like immediately after electroporation. So you can see the cells floating around in there. This is just to give you an idea of what it looks like immediately after transfection. You want to put your plate directly into an incubator at 37 degrees, just to make sure that the cells adhere to the plate and recover nicely. It's recommended that you leave your cells untouched in the incubator for overnight or 24 hours to make sure that they attach appropriately and recover nicely. I prepared an electroporation yesterday so that you can see what the cells will look like about 24 hours after electroporation, so I'll show you on the microscope. So you do see a lot of debris, but you can also see quite a few cells have started to attach. For instance, here you can see like the live cells have started to attach. That's a good sign your cells will recover nicely. So at this point, the final step would be to run an assay to determine your transfection efficiency. In the case of genome editing, you can do a genomic cleavage detection assay. This is where you'll PCR your targeted region, and then you will denature and reanneal your PCR product. If you have genome editing, this will create mismatches, and then you can introduce a detection enzyme which will actually cleave where the mismatches have occurred. I've prepared a slide which shows a GCD assay after genome editing using HPRT guides. You can see in lane 1 that there's one clear bright band, and there was no guide added to this lane or to this electroporation. So that band is showing you your fully intact product. In lanes 2 through 5, there are 2 clear bands. In these lanes, guide was added at different concentrations, and the 2 bands indicate that mismatches occurred during your assay, and this indicates a successful electroporation. So at this point, we've sort of wrapped up the training on neon electroporation, and if we have some time, I'd be happy to take any questions. Do we have any questions?
All right, thank you, Jackie. This is Eric speaking, and I will be working with Jackie through some of the questions that we have received online. So I'll try and divide them up in different kind of categories. So one group is more around the instrument itself. So could you speak a little bit more in terms of the difference between the 10 microliter tip and the 100 microliter tip that you mentioned?
Yeah, so like I said, there are 10 microliter tips and 100 microliter tip options. You would select which tip you wanted to use based on what's recommended in the protocol that you're using, which, as I mentioned, you can find on the Neon Cell database. You can also predict which tip you'll be using based on the number of cells you wish to transfect. Obviously, the 100 microliter tip can accommodate a much higher number of cells than the 10 microliter tip can.
All right. And so a follow-up question on that was that whether you could reuse these tips multiple times for multiple electrophorations?
So just as with any pipette tip, it's not recommended that you reuse a tip because you could cause cross-contamination. But in an instance where you might be doing duplicate transfections or something like that, you actually can reuse a tip. But it's not recommended that you use a neon tip more than 2 times in a row because the electroporation can actually cause thinning of the coating on the electrode of the tip, and this can mess with the conductivity of the tip and affect your transfections accordingly.
All right, thank you, Jackie. Would you be able to address how the neon system works with different cell types? Do you require individual buffer systems for the cell type that you use, or how does it work?
Yes, so the neon kit comes with a number of buffers. You can use, in this instance I use buffer R, and that's because I'm using iPSCs, but it also comes with a buffer T, which you can resuspend your cells in, but this is for blood-derived suspension cells only. So in most cell types you'll be using buffer R. And then in buffer E versus buffer E2, which are both supplied in the kit, buffer E is used with the 10 microliter tips primarily, and buffer E2 is used with the 100 microliter tips primarily. And I think there's a difference in osmolarity between the two, and they're optimized for the 10 microliter tip or the 100 microliter tip.
All right, that sounds really good, because that actually sounds like you can use one buffer for the majority of adherent cell lines.
Exactly.
All right, thank you. So now moving on, bringing the electroporation piece more into the area of pluripotent stem cells. So what are the kind of things that you can deliver with the neon very efficiently?
So as I demonstrated here, you can deliver CRISPR-Cas9 really efficiently. You can also deliver a number of proteins. You can deliver DNA. You can deliver siRNA, all with really high efficiency.
Okay. And how does that delivery compare to, for example, a lipofection reagent like lipofectamine stem or lipofectamine 3000?
So electroporation in general will give you a much higher transfection efficiency than other types of transfection such as lipofection, and particularly in instances like genome editing, you're really concerned with getting the highest transfection efficiency possible.
So with the electroporation though, are you concerned at all about cell survival post-delivery?
I mean, that's always a concern with transfection, and particularly electroporation, because you're putting the cells under a lot of stress. But I touched on this earlier, laminin 521 paired with our StemFlex media, and especially for StemFlex media, that will really up your cell viability post-electroporation. And then if you add Revitacell to your media, that will increase your cell viability post-electroporation even more.
Okay. Now maybe going a little bit more in the terms of protocols for the neon. Are there any specific protocols that you could use for pluripotent stem cells, or would we have to optimize for every single pluripotent stem cell line that we have?
So most cell types are, the recommended protocols are described in the Neon Cell database. So it's likely that you would be able to find your iPSC line in the database. If it's not in there, you can identify the cell type in the database that's most similar to the one that you're using, and then you can optimize, like that's a good starting point to do your own optimization from there, and you don't actually have to go through the full 24-well optimization, but that is an option as well.
So could you maybe elaborate a little bit more about the 24 conditions optimization, how you would go through that?
Yeah, so like I mentioned earlier, the device comes preloaded with 24 optimized protocols which you can load onto the device and perform your electroporation. The differences in each protocol is a difference in the number of zaps you're delivering and the voltage that you're delivering, things like that. And you can go through all of them if you desire and see which one gives you the best transfection efficiency and best survival for your cell type.
Okay, so you mentioned that several tricks to promote the survival of the cells after electroporation. Could you kind of ballpark how long it takes for the cells to recover and be ready for expansion and other experiments?
So if you have the most optimized protocol for your cell type, you should start to see your cells adhering to the plate and looking healthy after about 24 hours. And then your cells will grow at whatever rate your cell type tends to grow at, and then you can expand from there.
Okay. Going back to the Neon device itself, is there any difference in the efficiency between the 10 microliter tip and the 100 microliter tip, and would there be a reason in terms of efficiency why you would choose one over the other?
So I would definitely choose which tip to use based on what the recommended protocol says. But there's no reason to believe that there's a difference in transfection efficiency between the 10 microliter tip and the 100 microliter tip. Something to keep in mind though is that you do need to scale up your transfection, so make sure that your cell density stays the same. Also, if you do see that it doesn't work as well when switching between the two tips, you can adjust the voltage slightly, and that should correct for any differences that you see.
Okay. And now we have another question that came in regarding transfection efficiency. So would you be able to speak a little bit about, let's take the genome editing as an example, what kind of efficiencies that you're getting with genome editing after using the Neon as a delivery machine?
So the transfection efficiency you see totally depends on what you're delivering and your cell type and things like that. Typically you can see maybe between like 20 and 40% transfection efficiency, but again, it totally depends, and that's just a ballpark estimate.
Okay. So in terms of then using the Neon for genome editing, would you really recommend to use the Neon, or do you think that lipofectamine stem would also work?
For genome editing specifically, I would definitely recommend using Neon electroporation because, like I said earlier, in genome editing you're really concerned with getting a high transfection efficiency, and the Neon electroporation system will give you a much higher electroporation efficiency than something like lipofectamine stem.
All right, thank you, Jackie, for your wonderful demonstration. I'm sure it was informative for our audience, and I would also like to thank our audience for their active participation in this live session. Thank you very much, and I hope you enjoyed the rest of our 24 Hours of Stem Cells event.
Yeah, thanks for tuning in and joining me to learn about Neon electroporation. It's been fun.